Hello everyone, my name is Jasbir Kaur Simak. I've done my BTech in Biotechnology from MIT University, Mumbai, and my poster presentation topic is on molecular talking and toxicity analysis of FDA-approved drugs and plant metabolites against potential biomarkers to target human hepatocellular carcinoma. So, starting with the introduction, we know that cancer is the second leading cause of death globally after heart diseases, of which the most common types of cancer include breast cancer, prostate cancer, lung cancer, and liver cancer. So, hepatocellular carcinoma (HCC) is the one that accounts for the majority of primary liver cancer. That is on which my work is based on. So, it can occur. HCC can occur in patients that are already suffering from hepatitis B, hepatitis C, or cirrhosis, and some of the other causes of this disease include due to ingestion of aflatoxins due to excess alcohol consumption or due to metabolic disorders like NASH and NFLD. So the survival statistics related to HCC patients are very poor. About 2 years of survival is less than 50%, whereas 5 years of survival rate is below 10%. So why is the poor survival rate so poor? So this is due to the two challenges of HCC. The first one is in the diagnosis. So in case of diagnosis, the only biomarker that helps us in detecting if a person has got HCC or not is AFP, that is alpha fetoprotein. So, overexpression of this AFP could let us detect HCC. But the problem is that about less than half of the population that have got HCC, they have got poor, low levels of AFP or they are AFP deficient. So that is why early detection cannot take place, and most of the cases are detected at a very later stage. The second challenge is in the treatment options. So, the treatment options range from surgical resection to liver transplantation and also use of molecularly targeted therapy. The first two are very limited because they can only be used when the patients are detected at an early stage. So the patients that are detected at a later stage, they are left with only the use of molecularly targeted therapy that utilizes the FDA-approved drugs that include sorafenib, cabozantinib, regorafenib, and levantinib. But these FDA-approved drugs, they aren't specific as they are multi-tyrosine kinase inhibitors and they have got several side effects. The side effects range from mild, severe fatigue, diarrhea to severe night blindness, numbness, and blurred vision. And the other challenge in the treatment is that the resistance. So most of the patients that are treated with the sorafenib, that is the first line treatment drug, they incur resistance within 6 months of treatment period. So that is why uh, the survival statistics related to HCC are very low. And that is why we are looking for potential biomarkers as well as potential plant compounds that could help to overcome these two challenges. So, as per studies, we have chosen two potential biomarkers, namely transglutaminase 2, that is TGM2, and chromodomain and PhD finger containing one protein, that is BRPF1, because these two have shown their overexpression in HCC cells even when AFP levels were low or AFP was deficient. And the other is the natural plant compounds. So natural plant compounds, we chose that we have chosen them because they are known for their lower toxicity. They are known for their therapeutic and chemoprotective agents. Moving to the method. So first we chose the potential biomarkers. We that is BRPF1 and TGM2, and we downloaded their PDB structure from RCSB database. Similarly, for the ligands, that is the FDA-approved drugs, and the plant compounds, we chose them. We chose about 6 FDA-approved drugs and 157 plant compounds, and we downloaded their structures from PubChem site. And also, we had to convert the SDA format to the PDB one. Then we did the selection of the ligands using the PDB-Sum tool for the proteins. Then we had to perform molecular docking by the help of Autodoc Vina. So for this, we had to prepare several separate folders for each of the protein, for one for TGM2 and one for BRPF1. And then we, after docking with the help of Autodoc Vina Pol application, the compounds that had lower binding affinity to these two protein, they were then moved to the toxicity analysis by Vega QSAR. And then moving to the results. So in the first figure, you can see that the graph is against BRPF1 and the ligands. So the purple one refers to the plant metabolites and the blue bar refers to FDA drugs. So abisonon had the lowest binding affinity to BRPF1 of minus 10.6 kilocalorie per mole, followed by ephorphine and then cephranthine. As compared to the FDA drugs, the lowest affinity was with pimosite, followed by niclosamide and then regorafenib. So by this graph, we can see that plant compounds had better lower binding affinities to the BRPF1 protein. The second graph is against TGM2. So it is the same. The purple one refers to the plant metabolites, whereas the blue bar refers to the FDA drugs. So the first one that had the lowest binding affinity in plant compounds was camphorol, that had a binding affinity of minus 10 kilocalorie per mole, followed by cephranthine and then deutene. And if I have to compare it with the FDA drugs, cabozantinib was the one, uh, and cabozantinib and pimoside actually were the one that had the lowest binding affinity to the TGM2 protein of minus 8.7 kilocalorie per mole, followed by regorafenib. So by these two graphs, we can see that plant metabolites had better lower binding affinities to the potential biomarkers for both of the proteins. Then the top compounds were then listed to the toxicity analysis, and which we chose three tox model: mutagenicity model, carcinogenicity model, and in vitro micronucleus activity model. So this was done with the help of Vega QSAR. So we can see that most of the plant compounds were non-mutagenic as well as non-carcinogenic. Only ephorphine was the one that was mutagenic carcinogenic. And in case of in vitro micronucleus activity, four of them were active, and only routine had the inactive status in the case of in vitro micronucleus activity. So for this, we have to check the dosage concentration. And then moving to the conclusion. So with the help of molecular docking, we were able to find out that plant compounds had stronger binding affinity to the potential biomarkers, and abisonon was the one that had the lowest binding affinity to BRPF1, and camphorol was the one that had the lowest binding affinity to TGM2. It is also a good point to know that cephranthine was the only plant compound of the 157 plant metabolites that had the lowest binding affinity to both of the proteins. So that is a good point to note. And after that, when we performed analysis through Vega QSAR, we were able to find that almost all of the top plant metabolites they were non-mutagenic as well as non-carcinogenic, except for ephorphine. So our study concluded that plant metabolites may be less harmful and better choices for HCC diagnosis and treatment, and also further preclinical and clinical studies should be done to find out whether now for the confirmation of this potential biomarkers and plant metabolites. So that was for all for my presentation. I would like to thank my guide, Dr. Ashwani Sharma sir, for supporting and guiding me throughout my project work. And also, I would like to thank the organizers and the participating audience. Thank you, everyone. Have a nice day.